Hi, everybody. I'm BJ. I'm the genealogy librarian here at the Maine State Library. And tonight I'm going to start a series on genealogy and DNA. And what I'm going to do is I basically have what I want to cover, and I'm going to go fairly slowly and stop about questions periodically and go time wise, and then when it's time to stop for the night and then just pick up next week where we leave, because it may not, the dividing points may not be quite at the places I would do it from content. But I don't want to do too much and have you get overwhelmed. So, because I know many of us are not science people. And so if you are someone with a background in science, I've simplified stuff, and I know I've simplified stuff, but I want to make it understandable. So I tried to simplify it in a way that would work, so that people are seeing, are understanding what you need to understand in order to do the genetic genealogy side of things. Does that make sense to everybody? There are some things that I could go into more detail, but they don't really matter for what you're going to use the DNA for, so we'll skip them. So let me share my screen. And I will say that because of privacy concerns, there are places where I would rather do things live with the website, but I'm using the casino floor.
Using what?
Hey, whoever isn't muted, could you please mute? Thank you. I'm just getting everything set up here so I can see everything. But I'm presuming all of you can see my screen. Okay. All right. So yeah, as I was saying, I want to preserve privacy for people who match me, and so I'm doing this through PowerPoint with screenshots so that I can blur out people's names for reasons of anonymity. Okay, so let's get started. So genetic genealogy is the use of DNA testing with traditional genealogical research in paper records to construct a family tree, to connect people to each other, and to figure out relationships that may not be obvious in the paper trail. It does not replace traditional genealogy research. So if you think you can do a DNA test and be done with it, probably not the case. So anyway, what is DNA? Basically, it's the chemicals in your cells that contain the information about what you've inherited from your parents. And it controls things like eye color, hair color, or it has predispositions. People who are going to be tall if their environment allows them to have enough nutrition, things like that. So basically, what we're doing with genetic genealogy is looking at how similar two people are in what they've inherited from their common ancestors. And the more closely you're related, the more DNA you will share. That makes sense to everybody? Seems like a pretty simple statement. But a lot of people hear DNA, hear science, and if you're like me with numbers, your brain just freezes. So I'm going to go through this in the hopes that you'll understand enough that when we get later on and we're looking at some of the tools to manipulate your results, to use them in a way that will help you find relatives and ancestors, that you understand enough that it's not making your brain cramp. So, okay. So why do we do DNA testing? Well, for genealogical purposes, one thing is we can either confirm or refute a paper trail. One of the examples I'll probably use in the next week or two is a case where I have a fairly weak paper trail, and and there are couples who can possibly be the right couple in the late 1700s, early 1800s. And it turns out that I have a whole cluster of matches that are descended from one of those couples. And so it's let me say with much more certainty than I could with the skimpy paper trail that I'm pretty sure this particular couple are my several great-grandparents. One of the other things that people do is for finding family members. There have been several stories recently about people whose parents or grandparents were Holocaust survivors, and they've discovered second and third cousins. This also happens with those who are African American and people who were, third, fourth, fifth cousins separated by slavery. Sometimes you get previously not known family, often a sibling, or if a grandparent disappeared and had a second family. You get people who are doing DNA because they've either been adopted or that they know, say, a half sibling was adopted out and they want to find that person. And the other big thing is what they started calling non-paternity events, but more recently I've heard people much more frequently say not parent expected, for NPE, which means that in general, it's almost always the father. You get somebody who's not the parent expected. And it turns out that someone's father or grandfather is not who they thought it was. You also, and I'm not going to talk about this much tonight, but I will talk about it later in terms of testing and the variables in choosing which place to get tested. One of the both fascinating and most
realization about
most
intellectual preference.
Most concerning.
There are just so many options, and I still have to figure it out. So I actually just
Hey, Catherine, can you mute yourself, please? Or Rita, you're both okay. It's being used by law enforcement, and there are some real ethical issues, but there are also some really interesting issues coming out of this. And and I know I personally feel differently about identifying unknown crime victims and identifying criminal offenders, and those uses by law enforcement and the oversight of those. So we'll talk about that in a future week. But I wanted to bring it up that there are people who are concerned about it. And I want to talk about it so that if you have a relative who's concerned, you can answer their questions from a place of knowing the pros and cons. Oops. So, as I said, it assists with your genealogy research. It doesn't replace it. I've had people assume that they were going to just, spit into a tube, swab their cheek, and be done with it. The other thing, and this is sort of the last 2 points here, you don't, if you're not able to deal with a surprise, don't do it. If you're worried you're going to find out that your father wasn't your father and you're not comfortable with that, you maybe just want to don't do it. So that's something to bear in mind, that, and I'll talk more about that in a little bit, but I'm finding that, that when DNA testing started, everyone was talking about finding non-paternal events. And what I'm finding, the people who come to me at least, we're finding more that we're finding unknown, mostly half siblings, that a parent had a child before marriage or after a marriage, that occasionally during a marriage, that the other children didn't know about. I've had several people I've worked with where it turns out they are early baby boomers and they have a sibling they found from World War II era. So people born in the late 40s to mid 50s who were finding that one of their parents had a child in the early 40s that they didn't know about, with the various issues with the disruption of society during World War II. Men, mostly men in the military, young women going to work in factories in big cities. You mix a lot of younger adults and things happen. And then, he got shipped off or she went home to the farm because one of her parents needed help. And you end up with a child that later children of the couple didn't know about. So, and, sometimes you don't need a DNA test. You look at people and you go, I have one match that is an adoptive match, someone who was adopted out. And when she showed up in my matches, I knew exactly which side of the family she was from looking at her picture. I did not need to look at the shared matches to know which branch of the family she was. So that's sometimes happens. But I had to get a cat picture in. So let's look at this. This is an interesting case. This man named Joseph Newton Chandler died in Cleveland in 2002 with money in the bank, the better part of $100,000 in the bank. So local law enforcement decided that they needed to figure out who gets this money. And they looked at his paperwork and it said that he was born in Buffalo in 1937, which was all well and good. But when they looked a little further, they found that the Joseph Newton Chandler, who was born in Buffalo in 1937, died in a car crash in Texas in 1945. So if he died in Texas in 1945, he probably wasn't also dying in Cleveland in 2002. So that opened the question of who was he? And so the detective looking into this managed to find a tissue sample that had been taken at the time he'd had an operation a couple years before he died, that was stored at a local hospital. And at this point in the early 2000s, the only type of DNA that was able to be tested on a commercial scale was Y-DNA, which is passed from father to son. Fortunately, this person was male, so they could test that. And they discovered that there's a good chance that the people he was most closely related to through his straight paternal line had surnames that were on the range of Nicholson or Nicholas. And somebody's not, let me mute somebody here because it's really distracting. So thank you for staying muted. So the researcher who was looking at the DNA said to the detective, if you're looking at possible matches and you see somebody named Nicholson or Nicholas, look at them really hard because there's a good chance that it's going to help you figure out who he was. So later, what's called autosomal DNA testing came about. And through that, they discovered he was a man named Robert Ivan Nichols. Now, for some of you, this means nothing to you at this point. And I'm hoping when this series is done, if I went back through this story again, you'd be nodding your head saying, yep, I know how they did that. So that's why I showed you this little story, because it used both the early Y-DNA and the autosomal. And by the end of this series, you should understand how this was done. So there are 3 types of DNA that are tested. Mitochondrial DNA is passed from a mother to both her sons and her daughters. This is DNA that's actually outside the nucleus of the cell. And as I said, it passes just from a woman to her children. Then the other 2, the Y-DNA and the autosomal DNA, are in the nucleus of the cell. The Y-DNA is passed from father to son, and only men have Y-DNA. It's what makes men men, is to have the Y-DNA. And early testing was of this Y-DNA. And so sometimes I'll still get somebody say to me, well, I'm female, so I can't test. But autosomal DNA, which looks at the entire bit of your DNA, is passed from both parents to all of their children. And you get about 50% of your autosomal DNA from your father and about 50% from your mother. It's not perfect, 50-50, but it's really close. So here's a graphic of chromosomes blown way, way up. And humans have 23 chromosomes. If one of them mutates or is damaged, you get conditions like Down syndrome and trisomy 18, which means that trisomy 18 is a problem with chromosome number 18. And being scientists, they were really creative. And the longest chromosome is chromosome 1. The next longest is 2. Next longest is 3, down to 21 and 22 being much shorter than some of the others. So 22 of the 23 chromosomes are the same for all of the children of a couple, in the sense that you get a 50-50 mix, and you'll get one of these from your mother and the other one from your father. The last pair is the XY pair or the XX pair. If you're male, this pair is a Y and an X. If you're female, they're both X's. And so how it's determined whether you're male or female is whether you get an X or a Y from your father. So women have gotten an X from their mother and an X from their father. Men get an X from their mother and a Y from their father. Does this make sense to everyone at this point? Okay. So let's keep going here. So looking more at the types of tests, As I said, the mitochondrial DNA is from a mother to her children, and it is very slow to change or mutate, which means that the time scales it's working on are in the hundreds to thousands of years. And so for the most part, it's not that helpful at this point for genealogical timeframes where we're looking at a few generations. This is helpful to see where your straight female line ancestors were in the year 1000, but isn't going to really help you with somebody who was, living in 1883. As I said, you have Y-DNA. These are looked at, they do what they're called markers. They have other names, but markers is the more generic term. They started testing 12 and 25. Those are really minimal. You really need more to make good decisions about how related you are. So now the minimum really is 37. A lot of people do 67 or 111. The only company that does this is Family Tree DNA. And if you decide to buy a 37 or 67 and later want to upgrade, you can. They have the DNA and they will either rerun it or they've already run it and they'll release the data. As I said, this is father to son. It's your straight father's line. If you're female, you need to get a brother, your father, or a cousin through the male line to test. So an uncle's son. Autosomal DNA is those 22 pairs. It's measured in units called centimorgans. You really don't need to know what a centimorgan is. You just need to know the more centimorgans you share, and they test 7,400, and so you're going to get about 3,700 because you'll see in a bit, because you're matching a parent at 3,700. And you get chunks of centimorgans that are called segments. And this will be clearer after I go through a few more slides. And the more you share, the closer you get to that 3,700, the more closely related you are. It's also true that the higher the amount shared, the fewer possible relationships. So parents and full siblings are pretty clear. Then you get to the point where, aunt, niece, cousins, half siblings have some overlap. And that's where you will have to do some research if you find somebody you don't know. Same thing you get out to, is this person a great aunt versus a second cousin? And as I said, the 23rd chromosome is the X and the Y. And they do test your X chromosome. I'll talk more about that later. So here we go. So here you look at it, and this is a nice pedigree chart. And you have, this couple has 2 children, a son and a daughter. And the son has gotten his Y chromosome from his father's father, and so on back. And there may be occasional mutations, but for the most part, this is transmitted without change. And those small changes over a couple hundred years are what they're testing for. Mitochondrial DNA, they both got from their mother, who got it from her mother, who got it from her mother, from her mother. The daughter will pass on both her mitochondrial DNA and her X DNA to all of her children. Her sons will have a Y chromosome from their father, and her daughters will have an X chromosome from their father. But you'll notice, and this is, the Y-DNA was the early testing. And you see the problem here is that we are actually a combination of all of these ancestors, but this is only testing 1 out of 16, which is why they developed the autosomal DNA, which is testing all of these. And because of how things recombine, a good rule of thumb, this is one of the places where I'm simplifying, is that you're really only going to get back, for most cases, 5 to 7 generations on the autosomal DNA, just because it's been so chopped up by the time it gets further back that that's why it's hard to pinpoint, but there are ways to do it. And when I get to talking about third-party tools, we'll talk about how you can do, it's called DNA Painter, and how you can use shared matches to figure out that, I'm descended from this couple and so are you. And we can figure out, but another match is only descended from the wife instead of both. And so we know anything we get from her with that shared match is only descended from her and not the couple, had to have come from this person.
PJ,
Yes.
can you mute Catherine? It's distracting.
I can't. For some reason, it's not letting me. Okay, Catherine, it looks like you did. Thank you. Okay. So, yeah, I've been trying all along to keep people muted. I can see who is and isn't. So, anyhow, so the Y-DNA, as I said, they're informally called markers. They're technically short tandem repeats. And basically what they're doing is they're sampling at the same, a particular point, and they sample at 37, 67, 111 places. Autosomal DNA is measured either in centimorgans or percentage of shared DNA out of that. And we'll, next week when we talk about matching relatives, I'll show you more about that. So here we have, we have the Y-DNA that goes way back, the mitochondrial DNA that goes way, way back, and all these ancestors that aren't otherwise tested for. Whereas the autosomal DNA, I have a cat on the, come on, Scout down. No, that's better. Is testing what you've gotten from everybody, but is only going back 5 to 7 generations. So I said that's simplified, but it'll work for your what we need. But it's getting stuff from almost everybody. So here's a sample that is interesting. This is 6 generations of test takers. And so you have the person herself, and then her son was tested. And as you can see, they match almost perfectly because we get half of our DNA from each parent. Then this grandchild got half their DNA from this child and the other half from the child's partner. So at this point, remember, this is, they're testing, because of getting some from half, this is really 25% of the DNA, not 50. But, and this is the predicted amount by the theory. And this is the amount because what happened here is they actually got 22% from this person, which means they got extra from this person's partner. And then you see the great-grandchild got about half of that. And then each generation gets about half. And then this case of these centimorgans are in 24 chunks. Basically, it's 24 pieces of centimorgans. And this will be clear in a minute when I show you another graphic. In this case, you have the great-grandchild has 20 chunks. What's interesting here is the 3 greats-grandchild got a segment that looks like it just passed whole from his or her parent without doing any recombining or changing. And then you also get the small segments. So again, here you have the person who tested and then their child. And so, you have the green corresponds with this, and then this blue is the other parent, and we're not really talking about that. And as you go down each generation, you get lots more variation from the original. So here, parent, child, and each of these segments is the length of the whole chromosome. So these are chromosomes 1 through 22, and then here's the X chromosome. And then here's a grandchild, and you can see, so here on this chromosome, you have 2 segments, 2 chunks of centimorgans that this grandchild inherited from the grandparent. And here we have one where there's nothing. They got all of that one from the other grandparent. And then here, again, two segments, 1 segment, and so on. And if we look here, you have the parent-child comparison, the parent, grandparent, grandchild. And then when you go to the next step down where you have first cousins, you'll see, the segments tend to be shorter and there are fewer of them. So you have fewer centimorgans, fewer segments, and shorter segments overall. Does this make sense that the less related you are, the less you have in common? So again, what can happen, and I'll talk about this more when we're talking about some of the ethnicity stuff. You can get an ancestor here. Let's say this woman was Native American. Depending on how things recombine, you may not actually have any DNA from her. Or you may get an extra big chunk from this person over here. So that's something to be aware of, that for very small amounts of DNA, you will find that you don't match well, or you may have ethnicities that don't show up that you expect because they're just so small. And conversely, if you have something show up that's really small, it may be noise rather than actual ethnicity. I'm playing get the cat off the computer as I'm talking to you. So anyway, let's turn to Y-DNA. And usually I blot out the surname on a match, but because we're dealing with Y-DNA, which tends to follow surnames because of Western, because of European naming practices. I originally had my father test. It was actually kind of funny. It was almost 20 years ago. My mother was sick of what eventually killed her. And okay, Catherine, I see your back, and could you please mute yourself? I can't. Okay. And so I said to my father, I was up at my parents for Thanksgiving, and my mother asked what I wanted for the winter holiday gift giving extravaganza. And I looked at my dad and I said, you're a scientist, so would you be willing, there's this new technology to get your Y DNA tested. Obviously, I got an X from you because I'm your daughter, so I can't test. Would you do the test for me? It's gotten down, it's expensive, but it's gotten down into the reasonable price. And my dad looked at me and he said, does this mean I don't have to go to the mall? And I said, yep. And he went, sold. So I later had my brother redo the test because the company he did it with went out of business. As I said, only Family Tree DNA is doing these days. So anyhow, this is for 37 markers. You can see some of these say they've done 67 or 111. I've only done the 37. One of these days when I have the money, I'm going to bump up to 111. But, librarian salary. So anyhow, out of 37 markers, the closest I came are these 4 people who share 35 of the 37 markers. So what's happened is this company has taken the Y-DNA and basically spot sampled it in 37 places and said, you have the same thing at 35 of those 37 places. And that means that you are reasonably closely in a world scheme related, although for genealogical purposes, not very related. Now, one of the questions I had, most of you have probably heard me talk about how my great-grandfather was adopted. His name was originally McGee. And McGee is one of those names that can be both Native Irish Catholic and Scots-Irish Protestant in Northern Ireland, which is where my McGees are from. And so I was curious to do this to see whether they were Scots-Irish or Scots-Irish or Irish. And you go down these names and we've got Morrison, Fahey, Gallagher, O'Connor, McHugh, McCullen, Donahue, McGee, Donahue, lots more McGees right after this cutoff. Some Blaney's, some McCanns, names I saw all through my trip to Donegal and Northwestern Ireland. So yeah, Irish. You can put in your earliest known ancestor with that name. None of these are far enough back to start tapping into this distance. Haplogroups are basically saying this is a chunk of DNA that gets inherited together and we can trace where these people went. So I have a question in the chat about 37 versus 111 markers. The big difference is just kind of like seeing all of a photograph instead of half of it. You're just getting more detail. You're getting more points of comparison. So if it turned out that, let's say I did 111 and with this McHugh matched 3 at 111, that would show we're much more closely related than 3 out of 37. But it's giving us a clearer picture. Okay. And again, they tell you where the, again, this is at Family Tree DNA, and they're looking at where the earliest earliest ancestor for people who've done the Y-DNA is. And look at this, big surprise. 71 from Ireland, 26 from Scotland, and 13 from the United States. So even if I hadn't already known to look in Ireland, this tells me that's the first place to look. And here's why you want to do more. So this is a chart for 37 markers. And with a genetic distance of 2, there's a 50% chance that there's a common male ancestor in 6 generations, 90% in 12, and 95% in 14. But this is getting back to, 250 to 500 years ago, depending on how you define a generation, which, as those of you who are like me and have tried to do research in Ireland or in Ashkenazi Jewish families or in African American families, you're back before there are records at this point. Whereas this is the chart for 111, and it's showing you that if you've got a distance of 2, you're probably down to 8 or 9 generations or 8 to 12 instead of 12 to 18 for finding the common ancestor. And so you're into better odds at that point because you're getting a clearer picture. So again, here, we're at this point, 12 to 14 to 18 generations. And, I've numbered these from my nephew, but from my brother back to him, this is 6 generations, which is only 50% chance that those 4 people with a genetic difference of 2 are somebody I can tie into. And I'm not even sure if this name is right. Francis, the only reason I have it is it's on Francis's death record, and his kids were put into foster care when the oldest was 12. And so I'm not sure this name is right. I will probably be showing you next week or the week after. I've got a cluster that may help with this, but people are grabbing, there are too many Francis and Patrick McGee's that people are just randomly grabbing. So again, this is a chart. There are surname projects on Y-DNA. And so this is a case, these are all McGee's. There's a Morrison project. There's a McLeod project. There are various other projects looking at figuring out how everyone with a surname is related. And so what you have here, they're assigned a kit number to know who it is. Here's the name. Here's the oldest known straight paternal ancestor, where the person is, their haplogroup. And then these are the names for the markers where they have spot checked. And then the numbers, show you, for example, this person with the little purple numbers has mutations from all of these other people, but is otherwise about the same. And so they're lumped together. You can see here, this set of people, these 2 here are exactly the same. And so they're probably more closely related than the ones who have these. And if we look where I am, or my dad is on this, you can see here, this is actually a random sampling of McGee's that don't fit into other clusters. And notice how there are many more colored numbers here than on this one, where you've got clusters that are more closely related. And here you have more variations. And, my dad is probably more related to these people than these people, if it's a mutation that happened and they all inherited it. But then here's another case where the 10 is the, this 24 is the only one with all of these other 25s. And so when they spot checked here, they got a 14 instead of a 15. So you can see that these are sort of lumped together. And again, here's someone who only has the 12 marker. And so it's hard to tell because what if this person also has this mutation and this mutation and this mutation? It looks like they're two apart, but if they have all of these, but they haven't tested, they may be much further apart. They may be, 7 out of 67 rather than 2 out of 12. Does that make sense to people? Why the bigger number, you're able to get higher resolution on the questions of how related you are. It's generally not worth testing two brothers unless you're worried about the paternity for one of them. Like my father and my brother have the exact same results. It's probably worth testing first cousins if you're really stuck to see if they are the same. And second cousins really get, second and third cousins, if it's all the male line, really get you into the probably worth testing to see if there's a recent mutation. Because if there is, that tells you that it's like the mutation at your great grandfather rather than further back or closer. Whereas for autosomal DNA, it's worth getting everybody tested, and you'll see why, both with some stuff later today and some of what we do next week. I've tested my brother and myself at several places each in order to have better data. And so one of the things I'll talk about next week or the week after is the strategy for getting the most out of your money with relatives and at different testing services. So anyhow, I mentioned haplotypes briefly, and basically they're sets of markers on Y-DNA and mitochondrial DNA that tend to be inherited together. And so they're going to show a clump that works together. And you can sometimes, and this is where if you see reports of, we've traced the descendants of Genghis Khan or Charlemagne, it's because they have a haplotype that is found in a lot of their ancestors. And what happens is for those two people, and there's one that's called the Cohen modal haplotype that is for descendants of the biblical Jewish priests that they found from Jews from around the world, for those who have a family oral tradition of being descended from Moses and Aaron. They're going with the assumption that this haplotype they found in Central Asia, that is concentrated in Central Asia, and the further out you go, the fewer you have of them, that goes back to Genghis Khan because he was powerful and had a lot of sons. Same thing with Charlemagne. It may turn out that it was actually, Charlemagne's butler, but if rather than Charlemagne, but because the butler may have had more time to produce kids. But that's where you're getting it, is they're able to find signatures. And so you'll see this called a DNA signature or a genetic signature. And then haplogroups are people who have this common ancestor at the deep ancestry. We're talking 1,000 to 5,000 years ago, that they can trace as these groups of people migrate. And so this is getting into population genetics rather than genetic genealogy, but I figured I'd mention it because you're going to hear about it. And so this tells you things like, this haplogroup originated from a mutation that happened in Kazakhstan or the Basque country in Spain and France 2,000 years ago. And so we can't tell you any more than that. But if you've got this if you're part of this haplogroup, that means that your straight maternal ancestor was in that is descended from that person. So let's take a little more look at this. This is a comparison of me and my cousin Ginny. We both tested at 23andMe, and it's my dad and her mother are the siblings, so we don't share a mother's line. This is actually a cousin. I told the story last week with the newspapers of my father being a little clueless and not understanding that he was not the attraction and it was his younger sister. This is that sister's daughter. And so she gets her maternal haplogroup from my aunt. And so my father would have had this maternal haplogroup because both my aunt and my father would have gotten it from their mother. I got my maternal haplogroup from my mother, because this is looking at the mitochondrial DNA that we get from our mothers. And because Ginny and I are both female, you'll see that 23andMe makes no prediction about our paternal Y-DNA haplogroup. Now, this is a case, this is me and Ginny, and then the one on the right is me and Ginny's son, who also tested at 23andMe. And you'll see that he inherited his maternal haplogroup from his mother, who got it from her mother, my grandmother. And my father would have had the same one as these two people because he would have also gotten it from my grandmother. But then look here, you have Ginny and her son, and then this is Ginny's niece through a sister, same haplogroup. This is one reason this doesn't change. It's the other things that we'll talk about later that I'm really happy all of these family members tested at 23andMe. But it also makes a really nice illustration. These are all maternal line descendants of my grandmother and of my great grandmother. Sarah, on the other hand, is descended from Ginny's brother. And so again, she gets this from her mother, not from my grandmother or her mother. Does this make sense to you how this is inherited? This goes through women, it stops at men. Men get it, and it stops at a man. Because a man's kids get it from his partner, his wife. Now, this is interesting. I happen to have two second cousins, one on each side of the family, both named Scott. And obviously, they've been given their paternal haplogroup, which, again, I didn't get it because I'm not male. So it would be interesting to have my brother test there. One of these, I don't think either of these is a straight male. Oops, the cat is sitting on the mouse. Sorry about that. Neither of these is my father's, but they got these from, whoever their father's father was. And they can get pretty detailed. And the longer the number, the further downstream it is and the more detailed it is in general. So again, here, you can see they have this, so you have RM173, and then RM420 is a mutation off that. And then RM459 is a mutation off that. And then these are variants. you don't need to know more than this. I just wanted to show you because you will sometimes see the haplogroup, what it is. So yeah, this is my dad and my brother. And it's a variant that comes out of this. And, looking at this, this is each a stage where there's a mutation every circle. And so looking at this, it probably won't surprise you to see that this is most common. If you look here, it's a mutation off of this one. So it's probably not surprising that it's concentrated here and not over here, because this group of people had already migrated in this direction. And so for somebody to have a mutation that starts here, they're more likely to be in this area than over here. So see how that happens? And then obviously, these are the areas where there's heavy British Isles ancestry for this. And just to look a little more closely, this RM269 is one of the most common haplogroups in the UK, because it was a mutation they think happened somewhere in this area between Paris and what's now Belgium, the Netherlands. Even though it's empty here, they think it happened in this area, and then people immigrated that way. Oops, so let's talk. Okay, you know what? What time is it? Okay, I've been going for an hour. I'm going to stop at this point before I go into autosomal DNA, because I think I've thrown enough at you for this week. Is that reasonable? I want to go slowly enough on this that you can not feel completely overwhelmed. So let me stop sharing my screen. Where's the thing to do that? Okay, we're having like disappearing. There we go. And turn this back on. Okay. So I did this same program for my librarians group at 5 o'clock, and I got even less far with you guys than I did with them. But as I said, I'd rather go slowly and keep this to the hour than give you a complete headache from dealing with this. So yeah, this is going to be a 4 or 5 week session or series. But the basic things to get out of this are the more DNA you share, the more closely you're related. There are different types of DNA with different inheritance patterns that will tell you different things. And it can be a powerful tool to supplement traditional genealogical research. So I will be putting together a list of blog posts and books and other things I found really helpful for learning about this. And I'll probably send it out next week. The other thing is, if you start looking at stuff and have questions, I'm thinking what I'll do is first of all, I'll put the recording up so you can see that. I will probably also put the slides up at mainlygenealogy.com so you can go through and see them over the and look at them more. So anyhow, that's it for the presentation. Newspapers.com I've had like 5 people tell me, which is funny. I have a subscription, so I wouldn't necessarily pay attention, but newspapers.com is having a free weekend this weekend. So wipe the DNA stuff off your chalkboard and put up your newspaper questions for the weekend. And then we'll pick up the DNA again next week. So that's worth taking a look at because they have a lot of newspapers. So any other questions? Okay. Okay, so a question about a relationship from someone who's adopted. Larry, that'll come up next week or the week after. But feel free to send me screenshots of stuff by email. And maybe I can help you sort stuff out, or we can do a one-on-one at some point. And I can help you with you screen sharing. So I'll walk you through the specifics. And that offer's open to all of you. If you have matches that you aren't sure what to do with or what something means, feel free to make an appointment and we can do it. Either come into the library if you're local, or we can do a one-on-one Zoom and I can walk you through it and we can see if we can figure stuff out. You know, there's a lot of scary terminology on this. I will grant that. And I debated putting in about the haplogroups, but the thing is, if you start reading about this and looking at results, you're going to see these words. And I wanted to show you, you don't have to be afraid of them. So, you know, the haplotype is a set of DNA that gets inherited together. A haplogroup is a group of people who have inherited a group of these traits. And so you can sort of cluster them together. But it's looking, you know, the haplo stuff is looking at hundreds to thousands of years ago for the most part. Okay, so I've got some other questions. Let me take a look. Yeah, Roots Tech is free. Go for it. It's from Family Search. It tends to, at least last year, I haven't looked at this year's. Last year, they did a lot of like 20 and 30-minute programs. And a lot of you are going to go to a lot of their programs and say, oh, that's really simple, because a lot of it's aimed at new researchers and it's meant to be sort of flashy and attractive to people who aren't into genealogy. But, you know, it's free. You have nothing to lose but a little time. So I'd say, you know, sign up. If something looks interesting, go for it. And, you know, maybe you'll find it, you know, someone who does, you know, a better job than I do at, say, Kentucky or, you know, Georgia research. So go for it. So question, if you have Ancestry, is it worth getting one of the others like 23andMe? Often. And I will be talking in a couple weeks. We'll get to the point where I talk about how you decide where to test and what order to test in. Very briefly, Ancestry and 23andMe, you have to spit in a tube, swab your cheek. The others all do data uploads, and you can, for $20 or $30 instead of the $60 to $100, the test costs, you can unlock the data after you've uploaded it, after testing it, 23andMe and Ancestry. Ancestry has the fewest tools for manipulating your data, but they've got the largest pool of relatives. So especially if you're looking to solve a not parent expected adoption, something like that, you want to be at Ancestry. The other thing that happens is 23andMe does health traits, and they're the only ones who do that. And so a lot of adoptees or people who aren't sure who their father is or grandfather, other family, when they have a big hole in their family tree because of not knowing who a parent or grandparent is, they often do the 23andMe because it includes things like, do you have one of the genes that puts you at a higher risk for breast cancer or Parkinson's? And so a lot of adoptees have done 23andMe to get that first layer of the things where science has identified a gene where a variant has a health risk. But I'll cover that in much more depth. And it'll probably be in 2 weeks. I'll probably want to do it after I talk about looking at relatives. So I want to talk about how to play around with the relatives you found, and then talk to you about the testing strategy to get the best answers, to get the people in order to have the right relatives to test. I have to cover the autosomal DNA and the ethnicity part of it. We'll cover working with relatives and shared relatives. I'll talk about testing strategy. I'll talk about third-party tools, because there are some tools out there that are great, that are beyond what any of these companies offer. Some free, some inexpensive, and some expensive. And I probably will also take some time to talk about the concerns around law enforcement and how law enforcement uses data, and which data they can use and which data they can't use, and how that affects how you might talk a relative into testing, and how that plays into your testing strategy, and all of that. So don't be surprised if this takes me at least 4 and maybe 5 sessions. And I won't panic if we go into 6. Because I'd rather take the time to really explain it than rush through and have you feel like you've only got half the story. Does that seem reasonable? Okay, so let me stop the recording.